All right, so I'm gonna actually break this up into two separate screencasts. This first one, I'm gonna mostly just focus on turning on genes and gene expression in that way. And then the second one, once we turn on the gene and make the RNA, then we'll talk about what happens to that RNA to add so much variation in eukaryotes. So, part of this is the study of epigenetics. And when we talk about genetics, we're talking about the the genes specifically. But phenotypes are not determined just by what genes are present in the DNA of an organism, but by the expression of those genes. And when we talk about expression, we're talking about turning them on and turning them off, and how long they get turned on, and at what point in the development do they get turned on, because we can have the same genes in drastically different species and drastically different organisms, but how they actually express those genes can be very different. So, the study of epigenetics is not just focused on the genetic code or the genome, but how we actually turn things off and thus determine what the physical appearance looks like. So again, this time we're going to be talking about turning things on. The next video will talk about processing and processing the messenger RNA, how we actually do the translation, and then how we modify the protein possibly post translation, and then ultimately how do we turn off genes? How do we stop their expression? And we're going to talk a little bit here about turning them off ultimately before we even turn them on, which is kind of crazy as well. But part of this, we got to begin with understanding again the structure of DNA. So we have the code, right? But the code is bundled around these histones, and histones are the proteins that help DNA coil things up. If we are going to, before we can even begin thinking about translating a gene, we have to make the gene accessible. And what that means is we got to take this, if this is the little part of the gene here, we got to get that loosened up, because at this point, when the gene is wrapped so tightly around the histone, we can't actually begin transcription because none of those transcription factors can land, and RNA polymerase cannot land when these are so tight. So the first thing, protein synthesis happens because we loosen up the coils. And the big part of that is attaching these little things to little parts of the histone that hangs off. And we call this acetylation. So we're going to acetylate the histones and then expose the gene so that we can actually get the transcription factors to bind. So again, all of this comes back to proteins, and there are specific proteins called these histone acetylation protein, and this is going to be an enzyme that's going to ultimately help attach these acetyl groups to the histones, which cause them to loosen up. Now, there is also an enzyme that helps take off the acetyl groups, which coils it tighter. So acetylation is going to allow for transcription. Deacetylation is going to prevent transcription. So let's try to visualize that using this video.
DNA is packaged into chromatin. Chromatin consists of DNA, structural histone proteins, and non-histone proteins. Within chromatin, the repeating unit is the nucleosome. Nucleosomes are made up of 146 base pairs of 2 superhelical turns of DNA wrapped around a core of 8 histones. The histones are responsible for maintaining the chromatin's shape and structure. Epigenetic modifications, such as histone acetylation, occur at the amino-terminal tails of the histones that protrude from the nucleosomes. Acetylation of histones is generally acknowledged to play a key role in the regulation of gene expression. Histone acetylation is controlled by the balance in the activity of two enzymes, histone acetyltransferase, or HAT, and histone deacetylase, or HDAC. For a gene to be transcribed, it must become physically accessible to transcriptional machinery. Acetylation of histones by HAT causes uncoiling of DNA and an open chromatin structure. This causes genes to become accessible to transcription factors, allowing gene expression to occur and proteins to be made. Conversely, deacetylation of histones by HDAC results in tight coiling of the DNA and a closed chromatin structure. In some cancer cells, there is an overexpression of HDACs, an aberrant recruitment of HDACs, or an underexpression of HATs, resulting in hypoacetylation of histones and therefore a condensed or closed chromatin structure.
All right. So, we'll come back to here. Again, when we begin transcription, it's not so simple as, okay, we just need to find the start codon. We just need to find AUG, and then we can just begin transcription. The reality is we first need to loosen everything up. When things are loosened up, then we can have transcription factors that find the promoter sequence. And transcription factors are proteins. Those transcription factors are searching the DNA strand that isn't wound up, and it jumps over the tightly wound parts. It's going to be looking for this TATA box, because TATA is, the letters of the code, like TATA. And then it can bind to the promoter region. At that promoter region, we can get other transcription factors that join up, and then these activators, which again are proteins, and sometimes we often think of them as hormone receptor complexes, when they bind to the enhancer region. So when we talk about the regions, we're talking about the DNA code. This now can bend, and when all of this comes together, we then get RNA polymerase to actually begin at AUG for making the messenger RNA. So again, we're going to take a look at some videos to help understand this. We got to loosen things up, and then we have to get the transcription factors and the activators to the right locations on the DNA code before we can even begin any kind of transcription or making of the messenger RNA.
Characteristics. Muscle cells and neurons have the same DNA but perform different functions because they use different sets of genes. A key challenge in genetics research is to understand how transcription factors find the correct DNA sequences to control genes. In the nucleus, DNA is condensed into a DNA protein complex called chromatin. In places where genes are being expressed, there are often zones of naked DNA. We will now explore ways that transcription factors interact with these zones. Even among scientists, a common misperception is that transcription factors fall from the sky onto the DNA and test one spot at a time. Such a process is too inefficient. Instead, the transcription factor interacts non-specifically with the DNA backbone. This allows it to efficiently search the DNA for its binding site, a DNA sequence it recognizes. The transcription factor slides along until it encounters its binding site. Then it changes conformation to interact specifically with nucleotides on the interior of the helix. Now the transcription factor can stay and interact with other proteins for transcription. Or it can become dislodged by many factors, which could include movement of the DNA or interaction with other proteins. Studies have suggested that transcription factors sometimes move from one part of the DNA to another. Short hops might allow them to bypass other proteins or to hop over compacted sections of DNA. When the transcription factor lands on another segment of DNA, short hops become big jumps. Although the other strand is nearby in three-dimensional space, it may be much farther away on the chromosome, or on another chromosome entirely. There are 1,500 different types of transcription factors in human cells. They interact to create a complex language of gene expression.
All right, so again, we've got these transcription factors seeking out the right promoter regions, the Tata box and then the promoter region for specific things. Now, again, if things are coiled too tight, it's going to skip over those locations. Or if there is another thing bound to a certain location, it has to hop over that region. So if it jumps over that region where there is something bound to the DNA, that part can never get turned on. Or if it's coiled up too tightly, that part can never get turned on. But in addition to transcription factors and promoters, we also have to have the activators
Region
involved.
along the DNA. The length of DNA following the promoter is a gene, and it contains the recipe for a protein. A mediator protein complex arrives carrying the enzyme RNA polymerase. It maneuvers the RNA polymerase into place, inserting it with the help of other factors between the strands of the DNA double helix. The assembled collection of all these factors is referred to as the transcription initiation complex, and now it is ready to be activated. The initiation complex requires contact with activator proteins, which bind to specific sequences of DNA known as enhancer regions. These regions may be thousands of base pairs distant from the start of the gene. Contact between the activator proteins and the initiation complex releases the copying mechanism. The RNA polymerase unzips a small portion of the DNA helix, exposing the bases on each strand. Only one of the strands is copied. It acts as a template for the synthesis of an RNA molecule, which is assembled one subunit at a time by matching the DNA letter code on the template strand. The subunits can be seen here entering the enzyme through its intake hole, and they are joined together to form the long messenger RNA chain snaking out of the top.
Again, there is another region at the very end of that transcription unit or at the end of the gene that terminates the transcription. So again, as we're looking at this here, we have to have the transcription factors. Those proteins bind to the promoters, and then the activators have to bind to the enhancers. Then this needs to bend together, and when everything touches, then it can kick off the RNA polymerase, which ultimately then makes the code. Now, again, every single cell in our body has the exact same genes. So what makes one cell so much different than another cell? And a lot of it has to do with which genes actually get expressed. And when we begin with the initial fertilization, it is an absolutely kind of blank slate. But then over time, you have cells that differentiate and they become muscle cells, or they become um, skin cells, or they become liver cells. And over time, through that differentiation, you can have different things, these methyl groups bound to certain genes. So those genes get turned off. And this is not, again, this is epigenetic. So it's beyond just what the code is. It's beyond the genome. It's more affecting the phenotype. So if you turn off by methylating a certain group, you put a tag on it, those tags actually do get passed through mitosis. So if it is turned off, it generally stays off, and therefore different cells are going to make different proteins because different cells have different methylated areas. Now, the thing that is really interesting is it's not just um, the differentiation of cells that cause methylation. Methylation can be caused by environmental factors like stress and diet and smoking and all of these other things. They can add the tags and turn off certain sequences of genes, and those can be passed through mitosis. The other thing that makes cells different from one another is that they do contain different activators. So activators, again, can be hormone receptor complexes, but a liver cell is going to have different activators than a muscle cell. So if it has different activators, those activators are specific to certain genes. So you aren't going to you can only turn on the genes for which you have the correct activators for. So again, you can have all of this variation between expression and phenotype because of the different activators that are present, or through possibly the methylation. And again, methylation can hook onto different regions here. If you methylate something, you make transcription impossible. So it can prevent the binding of transcription factors. So if that transcription factor is running along and it's trying to find a location, if that location is blocked by a methyl group, then that transcription factor would jump over that area and keep on looking for the correct promoter region. So again, so far this video is meant to talk about how do we turn things off and start beginning this process of histone acetylation. The activators bind to the enhancer regions, the transcription factors bind to the promoter regions, and then when we bring all of this together, the RNA polymerase actually does the transcription, and it stops the transcription at the terminator sequence of the gene. From there, we now have this long messenger RNA sequence, and we'll get into processing.